Hi folks, this is David Villar and welcome to this third video on pesticides. Here we're going to discuss herbicides, which are by far the most widely sold and used pesticides of any group. Here is a figure showing the most common herbicides used in the US. This is a little bit old, from 2001, but it probably has not changed much to the present day. So on this video we'll discuss the three most common ones that, as you can see, are glyphosate, atrazine, and 2,4-D. One of the common ways to classify them is according to their mechanism of action. As we can see on the slide, there are many ways in which they work, such as mimicking plant hormones, also called auxins. They could also inhibit lipid or amino acid synthesis, or prevent the photosynthesis in the plant. On the right column, we list some examples for each of those mechanisms of actions. The ones in yellow are those that we will discuss next. And if you may wonder, there are at least 200 different herbicides that are commercialized in the US, with around 800 different brand names. We're going to start with glyphosate, which is the most popular one across the world. This is a non-selective herbicide, meaning it will kill just about every plant. On this slide, we see that it inhibits an enzyme which is specific for plants and is involved in the pathway of the synthesis of amino acids. And because mammals, we don't have this enzyme, the toxicity of glyphosate is usually very low or even non-toxic. So what happens to glyphosate in the environment? If we look at some of the physical chemical properties, it binds very tightly to soil, and even if it is highly water soluble, it is not likely to get into the groundwater because it prefers to stay in soil. In addition, the half-life in soils is just a few days, so it will not persist for a very long time after we do an application. And it is broken down very quickly by bacteria in that soil. And because it binds tightly to soil, you need to apply it directly on the plant if you want to be efficient killing that plant. In general, the acute toxicity is very low or non-toxic. As you can see here, the LD50 in rats of pure glyphosate is 5 grams per kilogram. That makes salt even more toxic. And if we compare it with caffeine and nicotine, we can see that there is a huge difference in acute toxicity. In most references, it will say that it is non-toxic to fish and wildlife, but there are always exceptions to that rule, and as we will see in a minute, for amphibians. And we should always keep in mind that killing plants may alter the animal's habitat, and that may cause indirect effects by disrupting food chains. So even if glyphosate is non-toxic to birds and mammals, the situation is different for aquatic organisms. As we can see here, it is moderately toxic to fish and aquatic invertebrates, but there are many amphibians that are very susceptible, and for which it would be very highly toxic on an acute basis. As we did, and we did also mention that glyphosate is very water soluble, and so if we apply it directly on a water body, it will reach concentrations that could easily be lethal to fish and other aquatic organisms. Now, the problem with EPA and other regulatory agencies in other countries is that they haven't required amphibian testing as part of their processes for registering most pesticides. And in fact, the herbicide has been on the market for nearly 20 years before the first amphibian study was ever conducted. The first experiments found that the commercial formulation of glyphosate, which is called Roundup, was much more toxic to amphibians than the active ingredient alone. So basically the toxicity was attributed to the surfactant, which, as you can see, is a polyethoxylated amine that we can see here on the slide. And these studies actually prompted a new label of not applying to weeds that were growing in or over water, and obviously don't spray across any open water body. What happens is that the same properties that allow the surfactant to penetrate the leaf cuticles also make it very particularly good at rupturing the gill cells of fish and tadpoles, and that's going to lead to their suffocation. On this slide, we see the LC50 for larval amphibians or tadpoles, for glyphosate-based herbicides that contain the surfactant that I just mentioned. Each circle represents an individual test on a different amphibian species, and the arrows indicate a worst-case scenario concentration that could be achieved on a direct application on a wetland or a pond, or you name it. As we can see in this figure, there are many species of frogs for which the concentrations can be reached in water with typical applications, and they are well within the lethal concentrations. Now the next one is on phenoxy herbicides that are a family of chemicals related to the growth of the growth hormone indole acetic acid. And when they are sprayed on broadleaf plants, they induce a very rapid and controlled growth in which basically the plants grow to death. 2,4-D is selective for broadleaf weeds. It will not affect grasses like wheat or corn. And when talking about this group of herbicides, the most infamous word, Agent Orange, always comes to mind. This was a mixture of 2,4-D and 2,4,5-T that was used as a defoliant herbicide during the Vietnam War. And in addition to its damage on the environment, the formulation was contaminated with the most toxic compound ever known, which is a dioxin, and that caused major health problems for many people that were exposed. So this dioxin, which is known as TCDD, or tetrachlorodibenzodioxin, was basically an unwanted product in the manufacturing process of Agent Orange, and it's also very persistent contaminant in the environment. One of the worst environmental disasters took place in a town in Italy, which resulted in the highest exposures of TCDD in a residential area. And the accident took place in a chemical plant that was producing some chemicals and herbicides. And there were at least 6 tons of chemicals that were released into the air, producing a cloud which was estimated to have at least 60 pounds of TCDD. So the population who lived in the path of this cloud developed acute signs like headaches, nausea, eye irritation, and hundreds of people had to be hospitalized for acute skin irritation, and many of those developed chloracne. As you can see here on the image, chloracne is a disease in which basically the people develop pimple-like eruptions of blackheads, cysts, pustules, particularly around the face, behind the ears, in the armpits, in the groin region. And the population affected that was followed for long-term effects had higher rates of different cancers and other immunosuppressive and endocrine diseases. We have to mention that phenoxyacetic acid has been reported to be toxic in dogs, most likely as a result of direct ingestion of the chemical, because in theory there would be very little exposure by simple contact on a lawn that has been treated chemically with this herbicide. So basically they need to be exposed to the concentrates or maybe some pools of the spray. And the mechanism of action is not clear, but there is a clear effect on muscle membranes that become depolarized all the time, and the animal shows myotonia. On this slide we see the title of some field and experimental studies in dogs. So we're going to describe one of them that took place in a Weimaraner 2-year-old dog that was presented with a 24-hour history of episodic, tonic, and extensor limb rigidity, a stiff gait. And when the animal attempted to stand, he would knuckle over the paws, and he would basically fall upon walking with all limbs extended. And the most sensitive effect of the toxicosis is found when we do a electromyogram. Basically, we're going to see repeated discharges that wax and wane. And they could not really identify a particular source, but the owner mentioned that the animal had access to a nearby plant nursery. They detected concentrations of 2,4-D in both the serum and the urine, which obviously confirmed the exposure. And the dog was only given IV fluids for 36 hours, and he made an uneventful recovery. Now, it's not mentioned on the study, but most often the only signs seen in field cases are just related to the GI tract, with vomiting and diarrhea, anorexia, and that usually precedes the rigidity of the skeletal muscles. They also mentioned that the animal had tics, but the signs of tic paralysis are different because there is a lower motor neuron disease. And instead of rigidity, the animal would have shown skeletal muscle weakness and decreased muscle tone. In both 2,4-D and tic paralysis, the animal maintains normal mentation, proprioception, nociception, and spinal reflexes. Next, we're going to discuss a herbicide that is considered as an endocrine disruptor. These are substances that disrupt the normal hormonal balance on any organism. For example, bisphenol A, dioxins, phytoestrogens, phthalates, DDT, PCB, and the one we're going to talk, atrazine. All of these substances, they have in common that they're very persistent in the environment, and at the very, very low concentrations, they can mimic or behave as if they were hormones, or maybe change their concentration in the body. We're going to talk again about frogs here. Atrazine has contributed to their disappearance in many countries. Frogs are good sentinels or indicators of a healthy ecosystem, and they play an important role wherever they live to keep that habitat in check or equilibrium. We say that they're very good sentinels because they're probably the first ones to disappear when a wetland is polluted. And part of the reason is because their skin is very permeable, and that is why they can breathe in the water, but at the same time that makes them very susceptible to any contaminant in the water. In general, they like ponds and lakes that are with very clean water, with slow flowing currents, and ideally with lots of vegetation that offer protection and a substrate to lay their eggs. On this other image, we see an egg mass in the water that will give rise to tadpoles when they hatch. Therefore, any substance that is in the water can also affect the viability of those eggs. By contrast, we as mammals, we harbor our offspring inside, and the placenta offers some protection from exposure to toxic substances. Here we see a pond in the backyard of a house. This is kind of the habitat that frogs like. We see a lot of aquatic plants that make it very appealing to frogs. And on this slide, we see the cycle that amphibians go through. The eggs give rise to tadpoles that feed on microscopic algae, and they look like small fish with their tail and gills that allow them to breathe in the water. So those tadpoles also keep the algae from blooming and create anoxic conditions in the water. And when those tadpoles convert into frogs, the gills disappear and they become lungs, so they can breathe out of the water. Those adult frogs now will eat and feed on insects that makes them very good predators of mosquitoes and flies that, as you know, can be a real nuisance. Here we see the tadpoles, which also serve also as prey for fish, so they become actually part of the food chain also. And there are 5 major enemies to frogs that are shown on this slide. Probably the number one and most important is the destruction of their habitat. for example, when we drain wetlands. Other ones are the introduction of invasive species that compete with them. Climate change is obviously changing the weather patterns, which is, for example, when it's supposed to rain, now it doesn't rain, and vice versa. And we have to mention environmental pollutants like atrazine. There can be a combination of all these situations, and that's what those arrows are pointing at, and they can all contribute to the population decline of frogs. Now, if we look at the physical chemical properties of atrazine, the water solubility is between high and low at 33 parts per million. However, this is enough because, as we will see, the concentrations of just 0.1 part per billion is enough to cause health effects in frogs. We could say the same thing about vapor pressure. It's very low, but just enough to carry enough atrazine at parts per billion with rainwater, and obviously contaminate areas that are far away from the original point of source. And the groundwater ubiquity score is high, and that implies that atrazine can contaminate groundwater. And the reason for that high value is because it does not bind to soil, and it has a very long half-life in the environment. In the US, in just about any water body, atrazine can be detected, even in drinking water. So how do we know that atrazine is an endocrine disruptor? Well, if you're going to remember anything on this presentation, it behaves as an estrogen, and whether frogs are male or female does not really depend on the microsomes, but the influence of hormones at the time of metamorphosis. And the frogs on this slide show the difference in color between males and females for one species. If we now expose those tadpoles at different types of estrogens, now all of them, all of the ones that should be males now become females. We see the natural estrogen, which is estradiol, and the other synthetic chemicals that behave like estrogen. And what is interesting is that when we expose those substances together with tamoxifen, the males will not become females. Basically, tamoxifen blows the activity of estrogen, and it is actually a drug that is used as an anti-estrogen to prevent breast cancer in women. In the US, atrazine, it has been #1 herbicide used in corn for many years, but now glyphosate has taken over that role. What happens to frogs exposed to atrazine? If we were to do a necropsy on frogs, that we can see that individuals become hermaphroditic. In other words, they have both testicles and ovaries, and those individuals are not fertile and cannot reproduce. And the mechanism of action is by induction of an enzyme known as aromatase that is going to convert testosterone into estrogen. Estrogen is obviously necessary to produce ovaries and develop the phenotypic characteristics of a female. And this study here, what they did is to expose tadpoles that have been recently hatched until they reach the state of metamorphosis. And they did so at concentrations of atrazine from going from 0.1 part per billion all the way up to 200 parts per billion. And they replaced the water every 3 days. And what they found is that at concentrations of just 0.1 part per billion, the individuals became hermaphrodites. And on top of that, the pharyngeal muscles would not grow, and those animals would not be able to sing. Only males are the ones singing in the frog world, and females stay quiet. And as we see on the figure, the levels of testosterone were very, very low in those hermaphrodite individuals. I would like to point out that what we are seeing here is very different from studies on the acute toxicity of a chemical, because here the effects are inside the animal, and we're not going to see any signs of toxicity, and basically those animals are apparently normal and healthy. On this other slide, they show what the reproductive consequences of atrazine were in frogs. the males were exposed to atrazine, and that basically caused them to be chemically castrated. And so here what they did is to put 4 females in contact with 4 control males and 4 exposed, previously exposed males to atrazine. And basically they knew what happened inside. Now they wanted to know the real life implications. And they found that the ones that were exposed to atrazine were not interested in mating with the females. And even if they did, they were not fertile, and the females would not produce eggs. So the outcome is shown in this other figure. As you can see, no successful pairings in animals exposed to atrazine, and very low fertility of those treated males. If we look at the testosterone levels, again, the levels were much lower. Normal animals have more than 5 nanograms per milliliter of testosterone, and the ones that were treated with atrazine were below that level of 5 nanograms per milliliter. When they sacrificed those animals, they show that many of them were hermaphrodites, and the testicles had almost disappeared or become atrophic. And the pictures on the left here show a normal testicle filled with sperms, and the ones on the right show those testicles with tubules that are now empty. So even if we turn those animals into a clean environment, they are going to be chemically castrated for life. On this other slide, we see the levels of atrazine that produce hermaphrodites in frogs, which are 0.1 part per billion, and the values that can be reaching water from runoff of a treated area or in temporary pools near treated areas, or even in rainfall water. As we see on the slide, it can reach levels of up to 40 parts per billion in rainwater. So as you can imagine, surface waters far away from the areas in which atrazine was applied can be contaminated with levels that will affect frogs. In this other study that was published in Nature, they took water from 8 different sites across the US, and they look at the proportion of frogs with gonad abnormalities. So they collected 100 frogs from each side, and what the microscopic image shows is that within the testicle there are many ovocytes. In other words, those animals are hermaphrodites. And as we can see in the legend, atrazine was detected in the water of all those sites at concentrations above 0.1 part per billion. That turns males into females, or males become hermaphrodites. The same thing in frogs is also taking place in other vertebrates. In birds and mammals, what it does is to reduce the production of sperms, but there is no sex change. The images on the left show normal testicles, and those treated with atrazine on the right are obviously empty. The concentrations of atrazine that cause a reduction in male populations is different in different vertebrates. For example, in frogs, it starts at 0.1 part per billion, and as we can see in fish, the concentrations start at 21 part per billion, and in reptiles, it also depends on the temperature. Concentrations of 0.5 parts per billion. If the temperature is higher, will cause a greater decline in the number of males. So if we were to summarize the specific effects of atrazine in frogs, we could say that it causes 3 major effects. One of them is demasculinization of males with empty testicles. Another one is partial feminization, that is basically testicles that have ovocytes. Or complete feminization, which is total sex conversion. As I said, birds and mammals, in birds and mammals, these feminization effects do not take place because it is the chromosomes that determine the type of sex. However, in humans, the consequences of inducing aromatase is also the conversion of testosterone into estrogen, and males basically become infertile. And the other thing that could happen is that the excess of estrogens makes the individual more prone to develop mammary tumors if it is a female, or prostate cancer if it is a male. The interesting thing about all of this is that the same company that manufactures atrazine also came along with a drug that prevents mammary tumors, or prevents them from growing any further. And as we can see, that drug is called letrozole. This other study was done in workers that were engaged in the production of atrazine, and as we can see, the incidence of prostate cancer is much higher, at least 8 times higher than the background population. With regards to the incidence of mammary tumors, it was shown that rats exposed chronically to atrazine at increasing concentrations of the chemical had higher incidence of tumors. It's difficult to extrapolate these findings to humans because we are not usually exposed to those high concentration. And as you can see, we're talking about parts per million, which is 1,000 times higher than parts per billion. Another effect that could be attributed to atrazine is birth defects. On this slide, we see that women exposed at increasing concentrations of atrazine in their diet or the water had higher numbers of children with birth defects. It is hard to make a definitive conclusion that atrazine is the real culprit, because these studies only prove an association instead of a cause and effect relationship. Now, the main effects in children appear to be defects in the formation of the penis and the testicles, and that's shown on this slide. Those defects are known as hypospadias, cryptoskidism, and micropenis. So this is the final slide, guys. Thank you for listening, and I look forward to seeing you on the next videos. So take care, and bye-bye for now.